So this is the first screencasted video lecture for biology this semester. This is chapter 1, the science of biology. What I'm going to do is take you guys step by step through all the most important content of the chapter to help you kind of focus your studies and what you should be considering to be the most important things, the things that I want you to emphasize in your studying and preparation for the exam. So let's first start with the breakdown of what biology is. Biology is the study of living things. So that would essentially be the study of life and all of life characteristics. So this goes all the way down to the smallest, most functional unit of life, which is the cell. All the requirements to support life, life characteristics, and the individual breakdown of all those different components. So I'll take you guys step by step through all of those concepts in this chapter and throughout the rest of the school year. Let's first start with a quick little rundown of what science actually is. So the mission or the goal of science is to basically understand the world. The world is always changing, and we are constantly trying to better understand the way that it functions, the way that certain things work, and in the process, you'll see the development of different laws, the acceptance and rejection of different theories and hypotheses, as well as using different types of tools such as graphs, diagrams, models, things that help us better understand some of the large-scale concepts in different sciences. When it comes to the scientific method, I think this is a concept that's pretty well understood at this point in high school. It's probably a set of rules and guidelines that you have become familiar with over many years of different science coursework. So one of the things you should be well aware of at this point is the difference between an observation and inference. So for an observation, we're talking about using your senses to either visualize or witness, something that is taking place. So it might be, using vision, the sense of sight, or it could be touch, it could be the sense of smell, description of a texture, or it could be, the sensation of feeling heat, things like that. Something has taken place. Now, an inference is a little different. An inference can be used or can be made actually from an observation, so they can go hand in hand. But an inference is basically a conclusion that is drawn, maybe something that wasn't witnessed but is now being seen. As in, I like to give the example that if a student dissects an owl pellet, they didn't actually witness the owl eating the mouse, but when they find the bones in the pellet, they can infer that the owl consumed the mouse as prey. So while they didn't actually witness the occurrence of it, they are able to actually draw a conclusion from what they have observed. And then experiments, you should know all the components to an effective experiment. So you have a control group, there's an experimental group, you should know the difference between an independent and dependent variable. We'll talk more about that in class. And the idea that bias should be removed from all aspects of science because we want to be as accurate as possible. And you want your results to be as dependable as possible. When you have accuracy and dependability, you're considered to be more credible, whether you're doing scientific research or any type of investigation. And then, of course, the use of the metric system. We need a commonly accepted system amongst all scientists and researchers so that they're speaking the same language and using the same basic numerical values, and there isn't any type of confusion between different groups. You should also familiarize yourselves with the microscope. Some really important parts you should know. The eyepiece, obviously, where you actually rest your eye to view different microscopes, whether it's wet mount or traditional microscope slide. You can see the eyepiece tube that connects to the eyepiece there to the head. And the head usually is rotational. You could definitely see that you have this like turret swivel effect where you can kind of move the eyepiece around. You have the nose piece or the objective turret. That's actually the part that's rotated so you can change the objective lens. And you can see where the objective lens is pointing to the right. Behind the objective lens, you have the frame. That's where you would actually hold the microscope with one hand. The other hand should be holding the microscope by the base at the bottom. You have two focuses, the coarse and the fine focus, depending on what type of setting, objective setting you have it on. So usually on the spanning or scanning microscope setting, you're going to just use the coarse focus, but at some point you're going to have to use the fine focus to really get a good resolution of what you're seeing. you want to, achieve clarity and be able to truly look at some detailed structures, you're going to have to use both focuses. Uh, Now you can see on the stage on the left-hand side where that's identified, that's where you set your microscope slide, whether it's wet mount or just a regular microscope slide. There should be at least one clip, possibly two clips, that allow you to stabilize your specimen or your different microscope slides. Then below that you have an iris diaphragm condenser, which is specifically designed for light to shine through. So this is a compound light microscope, and the illuminating system is below the condenser, and that's where the light is generated and shown through so that you can get light penetration through the actual object that you're viewing. So, and there should be some form of a switch or a knob that allows you to turn the light system on or off. Of course, in a compound light microscope, you need it to be on. So that's it for that one. Next, a little bit about data. So you have qualitative and quantitative data. The way that you can distinguish these two is qualitative data is describing different qualities about a set of data. These are going to be characteristics that don't utilize numbers. So if something can't be quantified, then it would be qualified. Quantitative data is using numerical values, so keeping track of different percentages or temperatures or population numbers, things like that. The reason we collect data is because a lot of scientific research involves experimentation and helps you keep track of all the results. And then once you have all your results, looking at all of your different conclusions. And doing so accurately will reduce your errors, so making sure you're keeping track of all the individual things that occurred during your studies, during the investigations and the experiments. You can see there's some graphs. You should be able to do some pretty simple graphing this semester for different population groups or tracking different types of temperature values and different things like that. So here are a couple of review questions that we'll be going over in class. You definitely want to write these down and make sure you're able to answer them thoroughly, and we'll definitely go over them in class. So section 2 is a focus on scientific attitudes and science in context. So there are 4 attitudes that pretty much are generally accepted by all scientists. You have an element of curiosity, which is just being thoughtful of the world, things that just naturally seem to intrigue humans. There should be an element of skepticism, not just believing something just willy-nilly if something is to be read or something has been told. There should be an element of doubt. It's just good skepticism to question it. There should be an element of open-mindedness, which basically should always leave the window open for opportunity to potentially find new answers or expand on current beliefs. And then the last one is creativity. Creativity is the reason why we have a lot of the answers to a lot of our scientific dilemmas in the past and currently. So we'll go over each of these in class. So when it comes to technology, science really is the main developing method for coming up with new technologies and developing ones that are better. Of course, not restricted just to scientists. You look at economics and you look at business, manufacturing, things like that. All of it depends on investment, and of course, as time goes on, we make improvements in technologies, and that's for citizens to use and people alike all over the world. Now, in terms of communicating results in science, the best articles, the best journals, the best texts, they're always peer-reviewed, and they're scrutinized and they're analyzed by scientists from all over the world. It gives them the opportunity to share different collections of data, draw different analyses and conclusions, which leads to the development of different theories, laws, and principles, which are considered to be scientific truth, well-supported, and they're just not basic premise. They're not just ideas that somebody believes strongly in. They're ideas that have been tested over and over again endlessly until they're considered to be accurate. So, and of course, those things can be questioned until the next best answer is found. So those are important things to keep in mind in communication of the results. Now, section 3 actually gets into biology. So biology is the study of life. It's the study of living things. And there are certain characteristics that all living things share. So number 1 is all living things are composed of one or more cells. Some organisms are unicellular, made of one type of cell, and other more complex organisms, more organized organisms, are multicellular. Every living thing has some form of genetic material that commands the cell to function properly. So this would take its form in either the molecule of DNA or RNA. We'll talk about nucleic acids later in the year. All living things need energy for growth and development, reproduction, maintaining homeostasis, just general functionality for their cells. So I touched on a few things there. And of course, all living things are subject to evolution. So, we talk about genetic differences and genetic changes and mutations. These are topics we'll cover this year, and the idea of evolution becomes paramount in the understanding of biology. All right, so big ideas. We'll start first with cells. So the basic unit of structure and function. All living things are made of one or more cells, and it's the cells themselves that actually have the organism function. So if something is going on with an organism, then something obviously is going on incorrectly with its cell functionality. The second thing here with DNA and RNA, which has to do with cells also, is the expression of genes and the expression of different traits. So whether it's like the traits of a human being with like eye color, hair color, or any other traits, the DNA or RNA is the commanding molecule that does set the functionality for cells. So those go hand in hand. All living things do need energy, and it needs to be constantly provided. Some organisms are producers and autotrophic. They can make their own energy. And of course, some are heterotrophs and they're consumers. So it could be decomposers, it could be active predators. But they, all living things have a metabolism that they have to support. So at a cellular level, they constantly need a supply of energy. And then this energy provides the ability for growth and development to proceed. So whether it's cell divisions that are making more and more cells to make the organism larger, or it's just, having development where cells undergo many changes and their functionality, and you get, the development of new traits and features. Next, we have reproduction. Two methods of reproduction are asexual and sexual reproduction. We'll talk more about that later, but it does provide the basis for organisms to reproduce. Then the survival of the species can continue. So reproduction is a really important idea in biology. Next, homeostasis. This is maintaining stable internal conditions, even though conditions outside of the organism or its cells may be different. So responding to different triggers and stimuli, allowing them to survive, whether or not conditions are, ideal, or if things are getting kind of rough, whether it's a dry environment or an oxygen lacking environment, which is anaerobic. Cells are constantly dealing with pressures in lots of different ways. We'll talk more about that later. And then number 7 is evolution. Over time, species have to evolve. It may be the environment that is putting pressure on the organism to evolve, or it's a set of mutations that maybe makes an organism better suitable for survival in a certain environment. The evolution will take place. And this definitely has to do with adaptations, traits that will allow organisms to, be better suitable for survival in certain environments. Now, certain individuals may have those adaptations and others may not. Depending on the environment, certain ones will have enhanced fitness. So we'll talk more about that in a later chapter. All right, next we have reproduction. So we have two types, sexual and asexual. Sexual reproduction pretty much just involves two different gametes. Gametes are egg and sperm. So the egg is fertilized by the sperm, which develops into a zygote, and then soon enough develops into an embryo, which eventually becomes, the offspring of the organism. So what's really notable about this method of reproduction is it pretty much results in offspring that are genetically unique, different from each parent, but, has genes from both parents. So while they are genetically unique, they will be similar to their parents, just not identical. So you can see how that works. You think about, different fertilization methods. There's different ways to do it, but it's the basic premise that the egg will be fertilized by the sperm. And then asexual reproduction, you just have a single parent. It's basically a mother cell which grows until it reaches its maximum size, and then it's basically programmed to divide. And in this process we call binary fission. The cell basically grows, reaches its maturity, its growth peak, and then it's triggered to divide into two genetically identical daughter cells. And it's It has its pros, it has its cons, obviously. With sexual reproduction, you get these genetically unique offspring, but in asexual reproduction, you get genetically identical offspring. The advantage is you don't need two different individuals, and this process is very quick. And in sexual reproduction, you do get the genetic uniqueness, but it means you have to find a mate or you need some form of pollination method. It's a much more complicated process. All right, this one here, the interdependence of living things, this has to do with ecology. Pretty much all living things require other living things for their survival. So the things that producers do allow for food production and different symbiotic relationships with heterotrophs and other decomposers and things of that nature. So no one living thing is self-reliant. They all rely on each other for their survival of their species. So that's something we're going to look at in ecology in chapters 3 through 6. And I think this is the last slide here. This is the definition of a species, a word that we're going to use periodically throughout the semester. Species is defined as a population or group of organisms that can interbreed and produce fertile offspring. So, you look at this picture here, those are polar bears. Polar bears are a species. And unfortunately, they can't breed with other species successfully. Now, they may be able to make viable offspring, but they have to also produce offspring that can successfully reproduce. So you think about a mule, which is the product of one parent being a donkey and one parent being a horse. Well, the mule is viable and it will live, but it cannot reproduce because its egg and sperm are sterile. So technically not a true species. Same goes for a liger, one parent a lion, one parent a tiger. But you can't make, or I should say, ligers cannot reproduce successfully. So ligers can't make ligers. The only way to get a liger is with one parent tiger, one parent lion. So we call these hybrids. They're not true standalone species. So these concepts that I covered in this lecture, this really brief one, just to introduce you to the ideas that we're going to cover here in the big sections in biology. So we'll be covering these topics periodically throughout the year. These 7 or 8 unifying themes are going to be all the topics that we cover in the textbook and subsequent lectures. So look forward to more of these recorded lectures and looking forward to a good school year. So thanks for watching, guys. Hopefully this was helpful. Let me know if you have any questions in the comment section below, and I'll see you guys in the next video.